During the process of DNA replication, what happens? One new DNA strand is formed. The production of new nucleotide strands is catalyzed by DNA ligase. Is it right? Production of new nucleotide strands is catalyzed by DNA polymerase. DNA polymerase. Initiation of the synthesis of new strand is brought about by primase. DNA dependent RNA primase. Okay, so stated is wrong. Short segments are spliced together by DNA polymerase. Does DNA polymerase bring about splicing? Splicing is done by spliceosome. Anyways, why do you do splicing of DNA? Splicing is a process that is seen in prokaryotic DNA. In prokaryotes, true or false? False. Prokaryotic genes are always polycistronic. True or false? Prokaryotic genes are polycistronic. Prokaryotic mRNA is polycistronic. Exons are not found in prokaryotes. True or false? Exons are not seen in prokaryotes. True or false? False. Introns are not seen in prokaryotes. So introns are non-coding regions. Exons are coding regions. Each new DNA molecule has one strand of nucleotides from the original DNA and one newly synthesized strand. That is correct. What is it called? Semi-conservative replication. Semi-conservative replication. Regions of DNA that do not code for portions of a protein are called. Okay, what are they? Anticodons, codons, exons, introns. Introns. Where do you find each of these? Where do you find anticodons? Where do you find anticodons? In the tRNA. Where do you find codons? On the mRNA. Exons. Exons and introns are a part of the cistron. Exon is the coding part, coding region. Intron is the non-coding region. Chemical reactions that convert glucose to pyruvic acid in the cell are called glycolysis. Tell me the beginning and end of each of these. Beginning of aerobic respiration and the end of aerobic respiration. The first substrate and the last product. For aerobic respiration, what is the first substrate? Pyruvate is the first substrate. Glucose. Glucose is the substrate. End product for aerobic respiration. Carbon dioxide. Glucose is completely broken down into carbon dioxide. Beginning and end of anaerobic respiration. Beginning is glucose, end is lactic acid. The end is lactic acid. Beginning is glucose, end is lactic acid. Okay. Citric acid cycle, beginning and end. Okay, because it is a cycle, you consider it as what enters and what exits. What comes into the citric acid cycle? Acetyl-CoA. What goes out of the citric acid cycle? Carbon dioxide. Glycolysis, beginning and end. Glucose and pyruvate. Glucose and pyruvate. Now for each of these, tell me net energy. Net energy, total energy. For aerobic respiration, total energy in terms of ATP, 38 ATPs. And anaerobic respiration, 2. Citric acid cycle. Number of ATPs, citric acid cycle for 1 molecule of glucose. Huh? How much? 24. 8 in glycolysis, 6 in the oxidative decarboxylation, 14, remaining is 24. 6 NADPH, 18, 2 FADH2, 22, and 2 GDT, 24. Glycolysis, 8. A sequence of nucleotides in DNA that provides a set of chemical instructions for making a functional protein is called a gene. Gene. Codon. Codon is how many base pairs long? What is the length of a codon in terms of base pair? Codons are 3 base pairs long. Anticodons are how many base pairs? Anticodons are how many base pairs? That is also 3 only, no? Anticodon is the sequence of bases which are complementary to the codon. 3. Anticodon is also 3 bases. Okay. Gene is the entire sequence. Microtubules are composed of actin filaments, essential components of cilia, flagella, centrioles and spindle fibers, responsible for changes in the cell shape, smaller in diameter than other cytoskeletal components. What is true for microtubules? Are microtubules made up of actin? Is the protein for microtubule actin? What is it then? Tubulin. Microtubules are made from protein tubulin. So it is not actin. Actin filaments form the microfilaments. Actin forms the microfilaments. So statement A is wrong. Though actin filament is also cytoskeleton, it does not make microtubules, it makes microfilaments. Microtubules are essential components of cilia, flagella, centrioles and spindle fibers. That is correct. Responsible for changes in cell shape. Do microtubules contribute to change in cell shape? They maintain the cell shape, but what is their function? Microtubules are contributing for intracellular transportation, movement of material from one place to another place within the cell. Microfilaments and intermediate filaments contribute to maintaining shape. Smaller in diameter than other cytoskeletal components. Microtubules are bigger in diameter. Microfilaments are smaller than this. An energy-bearing compound that promotes chemical reactions in cells and initiates the activity of metabolic machinery is what will promote chemical reaction and initiate activity of metabolic machinery? Energy-bearing compound. Now, NADP, NAD, FAD, CAMP. What is CAMP? Cyclic AMP. What is AMP? Adenosine monophosphate. Cyclic AMP's role is for intracellular messaging. It is the second messenger. Hormones will come and bind to the surface receptor. Hormones will trigger the cyclic AMP system. Cyclic AMP will go and attach to the genes in the DNA in the nucleus and stimulate the gene. That is the role of cyclic AMP. Cyclic AMP is second messenger. Cyclic AMP functions as a second messenger to what kind of hormones? What kind of hormones? Non-steroid hormones. Non-steroid hormones. Cell membrane. What is this? Nucleus. These are all DNA with genes. Now, receptor and receptor. Steroid hormone will cross the receptor go and act on the gene directly. Steroid hormone will cross the receptor and itself will go and act on the gene. Okay. Non-steroid hormone cyclic AMP will go and act. Now do you see this? When a non-steroid hormone comes and binds to the receptor, it will activate cyclic AMP, which is the second messenger, and that second messenger will go and act on the DNA to activate a particular gene. So steroid hormones directly goes in. Non-steroid hormones will act via second messenger system. That is all. Cyclic AMP is second messenger. So that is the one which will trigger metabolic activity. So initiating the activity of metabolic machinery means that. So the answer is cyclic AMP. NADP, NAD, FAD are all energy compounds. They act as electron carriers for giving energy to the metabolic reactions. The transfer of genes from one chromosome to another during synapses is termed as, easy, simple, what is it called? Transfer of genes from one chromosome to another chromosome? Crossing over. Crossing over. Linkage is genes being closer to each other, making them pass along together without scope for crossing over, depending on its proximity. If it is very close, crossing over becomes that much more difficult. The main function of the contractile vacuole in protozoa is nutrition, excretion, respiration, osmoregulation. Contractile vacuole in protozoa is for osmoregulation. An input of energy is required for which one of the following? Very easy. Diffusion, osmosis, active transport, facilitated diffusion. Active transport requires energy. Facilitated diffusion is also passive. Something will stimulate the movement of something else, facilitate the movement of something else. Gibberellin is obtained from, Gibberellin is obtained from Gibberella fujikuroi. Phytophthora infestans, late blight in potato. Fusarium produces toxin, but it doesn't produce gibberellin. Alternaria, early blighting tomatoes. Late blight in potato, Phytophthora. Alternaria, early blight in tomato. Which of the following theories gives the latest explanation for the closure of stomata? What is the accepted theory now? Starch hydrolysis was an older theory. When the starch hydrolysis happens, that will lead to accumulation of water in the guard cells and leads to opening of stomata. When the starch does not get hydrolyzed, it leads to placidity of the guard cells and closure of stomata. That was the earlier theory. Now it is movement of potassium ions altering the water potential within the guard cells. That is the accepted theory now. Munch theory explains phloem transport. Rough endoplasmic reticulum differs from smooth endoplasmic reticulum due to the presence of what makes the rough endoplasmic reticulum rough? Ribosomes attached to it. Which cell in the body, which tissue, the cells of which tissue in the body has a lot of ribosomes? Highest concentration of ribosomes is in which cell or in which cells of which tissue of the body? Nervous tissue. How do you identify this? Nissls granules. Nissls granules are ribosomes. Which of the following transfers electrons in the ET electron transport system? Quantosome, F particles, phytochrome, cytochrome. Which one contributes to transfer of electrons? Cytochrome. What are quantosomes? Quantosomes are associated with photosynthesis. They are the light harvesting systems. F particles, F0, F1 particle. What is it? F0, F particle is ATP synthase. On the inner mitochondrial membrane, you find some nails. Those are F0, F1 particles. Those are the ATP synthase complexes. Phytochrome is a pigment involved in photoperiodism. Phytochrome, pigment of photoperiodism, not associated with electron transport. Here it is cytochrome. In this, photolysis of water is useful for reduction of NADP, oxidation of NADP, oxidation of FAD, none of the above. Photolysis of water is required for what? See, technically, none of the options is the direct effect of photolysis of water, but without photolysis of water, one of these will not happen. Which is the one which will not happen without photolysis of water? Reduction of NADP, because NADP is the terminal electron acceptor, and if that fellow has to get an electron has to flow through the system, and for electrons to flow through the system, electron donor is water. After a very long time, I have started to exercise. Going 5 kilometers, walk in the morning, 5 kilometers in the evening. Today also I did. Day before yesterday when I was walking, somebody tried to steal my phone. Evening. One guy came very close in his bike. I was talking to my wife on the phone. I was holding it like this. He came very close. Suddenly I shifted the phone to the right side. I looked at him. He just passed by. It was pitch dark. I would not even have been able to see the phone number. I mean, not the phone number, the vehicle number, bike number. He was half my size, but there will not be such opportunities for confrontation. When somebody just pulls and steals. Maybe that is why he did not attempt to pull it from my hand. Because in that moment I just noticed that he is trying to do something. I would have pulled him out of the bike. He went away. Good for him, better for me. I have no interest in street fights, but Initially, I had asked, the gate of this campus is open, always it is open. Don't people come in and is there no issue with some stealing and all that? Somebody had told me, hey, no, nothing of that sort happens here. It's all very safe. So, you have to be careful wherever you are. There is a movie, Kannada movie, where the heroine goes to Europe. One comedian in that movie is settled in Europe for many years. He continuously talks bad about India and appreciates Europe. They are trying to keep their bag closed. He says, hey, this is not like India. You keep the bag, you come after 1 year, it will still be there. Have you seen this movie? And by the time they take a photograph and see, there is no bag. It's gone. The plants face wilting due to excessive fertilizer use because of what happens? You use too much of fertilizers, why do the plants wilt? See, too much of fertilizers will make the soil hyperosmotic. It increases the salt concentration in the soil, and this especially the inorganic fertilizers. Inorganic fertilizers destroy soil. I was going through a report of an ecologist, soil expert. So I think what he told was, if we remove the soil for many meters or half a kilometer or something, you remove the complete soil for many meters and replace it with good soil, it will still take many centuries for the soil to rejuvenate. We have already destroyed it. If we don't stop further destruction and start rejuvenation of the soil, it will not, we are not going anywhere. Bad times are ahead. We will die without food. Exosmosis. Exosmosis is what will cause loss of water from the plant into the soil. Exosmosis, endosmosis, imbibition, all three are different concepts. Exosmosis is movement of water from inside to outside. Endosmosis is from outside to inside. Imbibition is taking water on the surface. Can I say imbibition is similar to absorption? Is imbibition equal to absorption? Absorption is taking something inside itself, inside. Absorption is taking something on the surface. Which is a good absorbing agent? Absorbent which can absorb variety of things. You use it as a treatment for severe gastritis. In antacid syrups you use this. You use the same in water purification for absorbing micropollutants or microparticles within the water. EDTA is anticoagulant. All industrial grade filtration systems will have a very, have you seen that in those commercial grade water systems? You find that big blue color cylinder kind of a thing inside which something is there, this thing is there. What is that? Charcoal. Activated charcoal. Activated charcoal is a very good absorbent. So water purification, you pass water through activated charcoal, it will pull away a lot of impurities before you push it into the membrane filters and other filters for further purification. Right? So charcoal based water purification systems, you can just powder charcoal if you to make household water purification units, multi-level, multi-layer. You have some gravel, then smaller gravel, sand, and powdered activated charcoal. At the bottom of it, you have pure water. Gravel will remove all the suspended impurities, the bigger particles. Sand will catch hold of all the smaller particles. Activated charcoal will take out even further smaller particles, and you have pure water. And whatever you get at the bottom, you boil it and drink it. That's all. You don't need to have very expensive water filtration systems. So this unit will remove all the suspended particles and colloidal particles. Boiling will kill all the pathogens. Then the water becomes potable. The transfer of genetic material from one bacterium to another by a virus is called. We have discussed this. What is it? Virus transferring the genetic material to a bacterium. Transduction. Conjugation, transformation and transduction are the three means of transferring genetic material into the bacterial cell. Translation is not associated with this process. Translation is a part of central dogma of molecular biology. Translating the information or the code on mRNA into protein. In photosynthesis, what happens? Oxidation of carbon dioxide takes place, reduction of water takes place, oxidation of water takes place, reduction of ammonia takes place. What happens in photosynthesis? Oxidation of water. Oxidation leads to breakdown. Reduction leads to combining, building something. Origin of life was supported because of the reducing atmosphere in the prehistoric Earth. Reducing atmosphere allowed for complex compounds to be produced. Which of the following representations show the pyramid of numbers in a forest ecosystem? In a forest ecosystem, the pyramid of numbers is, in a forest ecosystem, the pyramid of numbers, can it be inverted? I'm not seeing the diagram. Yeah. Forest ecosystem, pyramid of numbers. There is one inverted pyramid. See, look at C. Look at C. What is special about C? By looking at C, what can you make out? What kind of an ecosystem is that? Huh? C. What? What do you notice there in C? Looks like a potato twisting from one angle. Spiral. That small narrow base is what? It can be a tree ecosystem, no? That can be a tree ecosystem. Rest all will be as per hierarchical numbers or reducing numbers. But one producer is tree. Forest ecosystem, what should we consider? We should consider B only. In a forest ecosystem, if you consider forest as an ecosystem, the number of producers will be more than the number of primary consumers and secondary consumers, goes on like that. Okay? RNA processing takes place in the cytoplasm, is the same as transcription, is an event that occurs after RNA is transcribed, is the rejection of old worn out RNA. RNA processing happens when? When does the RNA processing happens? Is it before transcription, or as the transcription is going on, RNA processing also happens, or RNA processing happens after transcription? Helicase is the helicase is unwindase, it will unwind the DNA. RNA polymerase II is associated with the transcription. Ribozymes are RNA molecules which catalyze chemical reactions. It is not a DNA synthesis mediator. One popular ribozyme, peptide transferase. Embryo culture is performed to develop seeds quickly, raise large number of plants, overcome dormancy and multiply, rare hybrids, avoid fertilization. In the previous paper also we saw embryo culture, you do it to protect the species, something that is threatened or something that has a risk of survival, you do embryo culture and propagate it. Overcome dormancy and multiply, rare hybrids. The enzyme commonly referred to as molecular scissors is, which is molecular scissors? Restriction endonuclease. Restriction endonucleases are invented for the purpose of genetic engineering. True or false? Restriction endonucleases have been invented for the purpose of genetic engineering. True or false? Restriction endonucleases have been invented for the purpose of genetic engineering. Those of you who say true, raise your hand. Yes, restriction endonucleases have been invented for genetic engineering. Those of you who say false. What are the rest of the people doing? Are restriction endonucleases created by humans? They are a natural enzyme, they are naturally present in bacteria. So they were not invented, they were discovered and exploited. Okay. When we discover something, the immediate consequence of the discovery is strategizing to exploit it. We develop strategies to exploit it. And you call that by the name human welfare. Okay. Exploitation of nature and anything that is not human is justified by human welfare. Restriction endonuclease cuts DNA at specific sites but does not destroy the cellular DNA because restriction endonuclease cuts DNA at specific sites but does not destroy the cellular DNA because the cellular DNA does not have cutting sites. The susceptible sites are masked by proteins. The restriction enzyme specific sites are modified by methylation. The restriction sites and the cellular DNA are in different regions. Why? Now the question is, of the same bacteria, restriction endonuclease cuts DNA at specific sites but does not destroy the cellular DNA of the same bacteria. That is how you should read it. If it can cut DNA, will it even cut off my own DNA? If I am the bacteria, I have produced the REN, restriction endonuclease, it can chop DNA into pieces at specific sites. If that site is available in my DNA also, will it chop off? Why not? It identifies the DNA fragment, no? It identifies the DNA palindromic sequence. So does it mean to say that there would not be that particular sequence in my DNA? Or should I produce a restriction endonuclease which will not have an identification site on my DNA? What? Prevention of restriction enzymes from acting on a particular site can be done by methylation of that base. You methylate the bases which are identified by the restriction endonuclease, the REN cannot act on it. So, it is creating its own shield, like stomach. Stomach produces acids which can destroy most of the living tissue, many living tissues. Then why does it not destroy the stomach itself? Because there is mucus protecting it. Pancreatic enzymes can destroy, melt organs if it comes in contact with organs. But why is the intestine not getting melted where the pancreatic enzymes are released every day? Protected by mucus. Methylation is that protection against the attack of restriction nucleus. Next, radial symmetry is usually associated with creeping mode of locomotion, lower grade of organization, sedentary mode of life, aquatic mode of life. Actively moving organisms will not have radial symmetry. Radial symmetry increases the possibility of interaction on all sides. So organisms which are sedentary or which have slow movement tend to have radial symmetry. Which of the following is transparent tissue? Tendon, ligament, fibrous cartilage, hyaline cartilage. Which of the following is transparent? Hyaline cartilage. Nothing is transparent. Translucent is the best way to put it. Hyaline cartilage is also not transparent. If it is transparent, how can you see it? It is smooth and glass-like, very clear. Huh? What? Fibrous cartilage is not transparent. Fibrous cartilage is opaque. It will have a lot of collagen fibers, which makes it opaque. Keratinized dead layer of skin is made of stratified squamous, stratified cuboidal, simple columnar, stratified columnar. Definitely not simple epithelium. So simple cuboidal and simple columnar ruled out. Is it stratified squamous or stratified columnar? Keratinized layer of the skin. Stratified squamous. Stratified squamous epithelium is formed on the skin. Collateral glands of cockroach help in copulation, fertilization, formation of ootica chamber, formation of ootica. What is ootica? Ootica is shell, egg case. Egg case. Alright? So collateral glands help in the formation of ootica or ootica chamber. What does it contribute to? Collateral glands. It contributes to the formation of the chamber, functioning of the chamber. Next. Periplaneta americana differs from Data orientalis in. Periplaneta americana is American cockroach. Blatta orientalis is oriental cockroach. There is racism in cockroaches also. They clearly divide who is oriental and who is occidental. American cockroach. See, generally humans also, Americans and Indians, if you look at, who is taller? Who is taller and more well-built? Americans or Indians? Americans are well built. They are very tall and more muscular than our people. Who is stronger might be something. Strength is relative. You cannot just gauge by looking at people. But when I say who is taller and more well built, Americans are more taller and well built. Likewise, American cockroaches are also more taller and well built. So they have well developed wings on the body. Blatta orientalis or oriental cockroach have shorter, less functional wings. So if you find some flying cockroach, do all cockroaches fly? All cockroaches have wings, but all cockroaches do not fly. Have you seen flying cockroach? Not like birds, they keep flying for kilometers. They are here, they have to move there, they can fly. If you see flying cockroach, that is American cockroach. If you see cockroach which is just running away, not flying, even in the face of grave danger, that is Indian cockroach. Shorter fellows. The long fellows, more muscular fellows, they are American cockroach. Urgent tapping cells are found in chapters which are out of syllabus. Trips of Liverpool. Trips of Liverpool. Trips of Liverpool secrete serotonin, brings about gut motility. Gut motility. Serotonin is a neurotransmitter also. Serotonin is a neurotransmitter as well. And serotonin is a hormone. Serotonin is a hormone which will increase focus, concentration. Serotonin's effect on the brain is increased focus and concentration. Serotonin is released when you exercise. Physical activity increases serotonin. Without serotonin, brain function reduces. I have at least about 50 to 60 students who left my school because children have to play every day. In our school, children do yoga one period, 40 minutes every day. They have sports and games for one period every day. Even the day before exams, the games period will not be cut. No teachers can take the games period for their classes. They cannot say my portion is take to complete and take games period. No. It is kept in a high sample. We say physical activity is more important than studies. If you have to choose between the two, be active physically, that is more important. Parents think otherwise, some of them say useless fellow, it tells children should play every day. Why are we sending them to school? You send them to your school so that you teach them. Why are you wasting time making them play? They will anyways play at home. Look at the unfortunate situation. They don't realize what they are doing. Children don't have friends outside of school. Children don't have place to play outside of school. And children are not allowed to play inside the school. Any of you have pets? Dogs at home? Pet dogs? Dogs have to be taken for long walks. Why? Otherwise the energy that they have, the intense energy that they have cannot be dissipated. If that energy is not dissipated, it will lead to anxiety issues, it will lead to psychological problems, it will lead to depression, restlessness. There is so much energy that is coming out from your system every day, and that energy has to be dissipated. It is like a borewell. If you have a borewell, if you keep using it, the source of water continues to remain. You don't use that. You dig a well, you dig a borewell. You have got access to a stream of water underground. You keep pumping it periodically. The water keeps flowing. Otherwise, nature has a way of blocking that path. It is a leakage. It is underground current. It's all underground rivers that are flowing. If you have made a hole into that, nature has a way of blocking that if it is not flowing. Same with this. There are millions of kilometers of energy flow in the body. You don't let that flow, it gets blocked. And people die very soon. I say our education system is killing children even before they turn 18. Beyond that, it is just waiting for funeral. It is such a grave situation. It is destruction of an entire nation. You look at China. Chinese have a lot of bad policies and all. That apart. They are very expansionist. They want to conquer the world. They have bad economic practices where they put people into debt trap. But look at their system. Look at the games they play. Our children are doing reels. They are playing play, art, sport, everything they are exposed to. Look at Japan. Who listens to me? I have got my weapons. Who are my weapons? I am weaponizing all of you. Okay. Put these thoughts. Hope that somebody somewhere will pick it up and take it further. That's all. That's all you can do. You have to keep doing what is right for your entire lifetime. Somebody will get inspired, somebody will join you. That doing right should not die. You should not stop it. Somebody should keep doing it. Alright. System will change when there is enough, when the critical mass attains, when the change attains a critical mass, that becomes the system. You will see. Stenson's duct is associated with, again leave it off, but Stenson's duct is associated with carotid gland. Stenson's duct. Such beautiful ducts. You should forget them now. Stenson's duct. Wharton's duct. Ducts of Rivinus. Sounds like music. Which one of the following statements is wrong as far as symptoms of emphysema? In emphysema there is increasing breathlessness. True or false? In emphysema there is breathlessness. Why there is breathlessness? Because of the loss of elasticity of alveoli, the gas exchange efficiency reduces and that leads to breathlessness. The patient may have difficulty in walking across a room. True or false? If gas exchange is not happening properly, when the patient starts walking across the room, demand will be more than what the patient is able to supply. Taking the air in inspiration becomes difficult. True or false? False. In whatever condition, in any of the COPDs, chronic obstructive pulmonary diseases and asthma also, inhalation is more comfortable than exhalation. Exhalation is more difficult. Why? Why? Consider this. Let's say this door is the passage for air. This door is the passage for air. When you have to enter the room, I am pulling the door wide. For you to get out of the room, I am squeezing the door. Which one is easier for you? To come in or to go out? For inhalation, the respiratory tree is stretched. There is expansion, there is pull. For exhalation to happen, the respiratory tree is compressed, making the tubes narrow to increase pressure. Now this passage, if it, let's say you are of the size of the door. You are of the size of the door. When you have to come in, when you are trying to come in, the door gets stretched. When you are trying to go out, the door gets pressed. Which one becomes difficult for you? Coming in or going out? Going out becomes difficult because the door becomes small and you have to still go out. Your size doesn't change. The volume of air coming in and the volume of air going out is the same, no? 500 ml. Inhalation is also 500 ml, exhalation is also 500 ml. That is tidal volume. But when you are inhaling, the size of the lumen of the vessels or the passage, the respiratory tree, becomes bigger because you are stretching the capacity. You are increasing the volume. For exhalation, you are squeezing it to increase pressure so that air gets out. It has to get out to a narrower tube than what it was. Now in that, if you have some obstruction also, if you have some boulder in between, then that is COPD. If you have a table, the table is there at the door, that is COPD, obstructive disease. Inhalation also will be difficult for you because it has become narrow at that point. But exhalation will be more difficult. Yes or no? Next. Difficulty in exhaling air out during expiration, that is correct. Huh? Correct option is C. Huh? What? Correct option is option C only. Because you have to find out which is the wrong statement. The other three statements are right about emphysema. Okay. The cardiac or heart muscles beat always rhythmically by external stimuli, their own property, action of liver, action of hormones. Their own property. What is that property called? Auto-excitability. Heart muscles have auto-excitability. True or false? Heart muscles have auto-excitability. True or false? Because heart muscle, sinoatrial node and atrioventricular node and all the fibers conducting system, all of those are heart muscles only, cardiac muscles only. But can they fire independent of the nervous system? Can the cardiac nodes fire independent of the nervous system without stimulation from the brain? Can heart function? Can it function? So if I remove the heart, detach it from the brain, detach it from all the nerves, provide it, keep its blood supply intact, I'll do a dissection of the heart, I'll stun the person, I'll do a dissection of the heart. This is what we used to do for frogs, to test the effect of drugs, sympathetic stimulation and parasympathetic stimulation on the heart function. We used to do this experiment. We had a big frog tank in our pharmacology lab. Frog tank. You have to go and catch hold of the frog. You have to go, identify a frog that you want to kill, catch hold of it. In your non-dominant hand, you will hold the frog, bend its head. So access the base of the skull. And in our dissection kit, there used to be one pin, a big pin, rod kind of a pin with piercing tip. Puncture it and destroy the spinal cord. You destroy the spinal cord. When you destroy the spinal cord, what happens? You just push it and you just keep rotating this pin. It destroys the spinal cord. The entire peripheral nervous system is destroyed. When the peripheral nervous system is destroyed, muscles are paralyzed. The frog is no longer able to use its skeletal muscles. So it is paralyzed. But it is living because the autonomic nervous system is still there. Autonomic nervous system is not destroyed. So carefully you will dissect the body of the frog. You will remove the heart. You don't damage anything else. You remove the heart, you dissect the heart, you mount it on a pin which is attached to a liver which has a pencil to it, and the pencil is touching a graph paper. So the heartbeat will be transmitted to the liver and it keeps on marking on the paper. And the paper is on a rotating drum. So the drum is rotating, the heart is beating, pencil is marking, and it keeps rotating. It's set at a particular speed. When you add a sympathetic drug, heartbeat becomes faster. When you add a parasympathetic drug, heartbeat becomes slower. So don't worry, it is bad now. You don't have to do that frog dissection for your pharmacology experiments anymore. Now my question is, can heart operate without nervous system connection? It can't. Sympathetic and parasympathetic system will determine how many times these fire. The cardiac muscles are getting impulses from the nodes which are cardiac muscles. The impulses are produced by the cardiac muscles only. But the nodes have to be told how much they should fire based on the nervous system stimulation. Understand that? So their own property. Aldosterone hormone controls sodium ion reabsorption. Lower levels of aldosterone secretion cause what? Lower levels of aldosterone secretion causes sodium reabsorption will be affected. Sodium excretion will happen. Increased excretion of sodium ions. What will that lead to? If there is increased hypoaldosteronism, what will it lead to? Hypoaldosteronism. What might happen? Excessive sodium loss. Electrolyte imbalance can happen. Electrolyte imbalance can happen, and sodium is not only required for blood pressure. Sodium is required for nerve functioning. No, the nerve impulses happen because of sodium. So sodium reabsorption getting affected means the nerve impulse nerve conduction will get affected. Substances like pigments, antibiotics, hydrogen ions, urea, etc. are excreted mainly by tubular secretion, reabsorption, both A and B, none of these. See, these options are very simple options because both and none, you leave it out. Tubular secretion and reabsorption are opposites. Correct? The question is asking about excretion. Reabsorption cannot do excretion. If you are reabsorbing, you cannot excrete. So even if you don't know by common sense logic, you can still be able to answer this. Tubular secretion. Secretion into the tubes. Tubular secretion happens in which part of the nephron? Secretion happens in which part of the nephron? Mainly for hydrogen ions, antibiotics, pigments, all these things are excreted by tubular secretion. Tubular secretion is into which part of the nephron? DCT, distal corollary tubule. A person taking a protein-rich diet will eliminate more urea, creatinine, albumin, glycogen. High protein diet will lead to excretion of what? More excretion of what? Urea. Urea is a safer metabolite. Creatinine is related to muscle metabolism, not protein intake. Creatinine is related to muscle metabolism. Albumin is blood protein, not excreted. Glycogen is a carbohydrate. It is a complex carbohydrate, storage carbohydrate. So it is between urea and creatinine, you should know. Urea is directly related to protein metabolism. Creatinine is associated with muscle metabolism. Bodybuilders take supplements with creatinine in it. Creatinine supplements they take to enhance muscle metabolism. With enhancement of muscle metabolism, the muscle build-up will be faster is the logic. But unless you are preparing for some bodybuilding competitions, weight training for fitness and a good lean body should not include any supplements, protein supplements. You should have dietary protein. Supply your requirement through dietary protein, not through ready-made supplements. It has its own counter effects. And if you are training for bodybuilding, then supplementation would be required, and supplementation should be under the strict guidance of experts. Not gym trainers. A bodybuilder who is a gym trainer may not be an expert in nutrition. It should be an expert nutritionist and doctor's advice. Autonomic nervous system, which operates independently from the brain, does not regulate. ANS does not regulate respiration, blood circulation, excretion, learning and memory. What doesn't it control? ANS. Learning and memory. Learning and memory are exclusively higher order skills, higher order functions of the brain. Terminals of the post-ganglionic fibers in the sympathetic division secrete myelinated axons. Preganglionic fibers are myelinated. What is the advantage of myelination of a nerve fiber? Fast conduction. Fast conduction. Injury to the adrenal cortex is not likely to affect the secretion of which one of the following? Cortisol, aldosterone, both aldosterone and dihydroxyepiandrosterone, adrenaline. Adrenaline. Why? Adrenaline is the adrenal medulla hormone. It is not the cortical hormone. Cortex produces glucocorticoids and mineralocorticoids. In the given options, cortisol is glucocorticoid, aldosterone is mineralocorticoid. Which of the following hormones is not a steroid? Androgen, aldosterone, testosterone, vasopressin. Vasopressin is a peptide hormone. Androgen is testosterone, estrogen. These are steroid hormones only. The zygomatic arch bone is found in the zygomatic arch is where? Where is the zygomatic bone? Where is the zygomatic bone? Cheekbone. Cheekbone is zygomatic bone. Zygomatic arch is this here, which forms the under eye ridge. Okay. So it is in the skull. Vertebrae, sternum, pelvic girdle. No, right. In most mammals, vertebrae are. In most mammals, vertebrae are acellus, procellus, heterocellus, platypocellus. Acellus means both the surfaces are flat. Both the surfaces of the vertebrae are flat. Prococellus is concave in the front. Heterocellus is saddle-shaped vertebrae, and platypocellus is, concavity on both sides. Alright, this is beyond what is required for you to know. No need to bother about this question. We don't have to go so much deeper into the shape and the further classification of the vertebrae. Joints are important in the skeletal system, not the different classifications of different bones. The axis of a vertebra acts as a centrum of the atlas. Transverse, articular, odontoid, palatine. Odontoid process of the axis is the one which acts as a pivot to the atlas. What type of joint is this? What type of joint? Pivot joint. In sweet peas, genes C and P are necessary for color in flowers. The flowers are white in the absence of either or both genes. What will be the percentage of colored flowers in the offspring of the cross between this? What kind of cross is this? What kind of an interaction is this? What kind of an interaction is this? Complementary genes. This is complementary genes. That means what? A character is expressed when both the genes are present positively. Alright. Now let's say capital C with capital P. What is the probability? %25. 1/4. Correct, no? Capital C with capital P, probability is 1/4. See, for calculating that, you don't have to do the entire cross. This is the varieties, no? We want this with this. We want this with this. So, 50% of capital C with this capital P and 50% of capital C with small p. Capital C with small p, we don't bother. So this gene will give us, okay, let's say capital C, small c, small p. Here it is, small c, capital P, small c, small p. Well, no, these are the four. These are the four possibilities. We'll do it off. We'll do off the. So this will give us this capital P, capital C, capital P. This will give us, this is capital C, capital P. This will give us, right? Let's do off a proper cross. Okay. What are the genotypes here? Capital C, small p. This was capital C, small p, no? I have written capital P. And small c, small p. This one has two types of gametes. This is small c, capital P, and small c, small p. Capital C, capital P, yes. Capital C, small p, no. Small c, capital P, no. Small c, small p. Out of the four possibilities of genomes that are possible, there is only one which will have dominant allele for both the genes. Dominant allele for both the genes. Even if you do the entire cross, you have two types of gametes possible for this individual and these two types of gametes possible for the second individual. Proportion will remain the same. You will get 16, but proportion will still remain the same. So 25% of them will be colored. If a homozygous red flowered plant is crossed with a homozygous white flowered plant, the offspring will be. If a homozygous red flowered plant is crossed with a homozygous white flowered plant, the offspring will be half white flowered, half red flowered, all white flowered, all red flowered. Now looking at the options, you have to assume that this is a Mendelian cross. So all of them will be red colored because this is a first generation cross. Mendel was not able to say anything about recombination and crossing over because he did not have a large and strong microscope. He chose only pure types. The traits he chose were not linked and present on different chromosomes or were far apart. The traits he chose had no genes. Fortunately for Mendel, he did not come across his characters were not linked characters. They were all present on separate chromosomes. That was the luck Mendel had. Albinism and phenylketonuria are disorders due to recessive autosomal genes, dominant autosomal genes, recessive sex-linked genes, dominant sex-linked genes. They are not sex-linked, so both recessive and dominant sex-linked ruled out. They are recessive characters, autosomal characters, recessive autosomal disorders. Which plant species is on the verge of extinction due to overexploitation? Gloriosa, Podophyllum, Ceratella. They are all medicinal plants. All of these apparently are on the verge of extinction. If high altitude birds became rare or extinct, the plants that may disappear along with them are. Which plant might disappear if high altitude birds will become rare or extinct? So it would be plants which are growing at high altitudes and are dependent on birds for pollination. Which of these are? Rhododendrons. Rhododendrons. If the Bengal tiger population becomes extinct, what will happen? Hyenas and wolves will become scarce. Why? Why hyenas and wolves? They are independent, they will be happy, they will thrive because they will have more food to eat. The wild areas will be safe for man and domestic animals. Its gene pool will be lost forever. The populations of beautiful animals like deer will get stabilized. Look at the option. The populations of beautiful animals like deer will get stabilized. Instantly villainizing the Bengal tiger. Telling that it is because of the Bengal tiger that beauty is not able to survive in the wild, in its eating. Gene pool will be lost forever. Anything that is extinct would lose its gene pool. Which of the following is a vestigial structure in python? Hind limbs, teeth, poison gland, scales. Hind limbs. The remnant of the hind limbs as a pelvic spur, some small extension from the tail end is present in snakes. That is the vestigial remnant of the hind limbs. Acting as an evidence for our friend Lamarck's theory. Which of the following monkeys have a prehensile tail? What is prehensile tail? Tail which can be used like a limb. Have you seen some monkeys hanging by the tail? They will use the tail to coil around branches and they can hang by the tail. Use the tail like limb. Which monkey has that? Spider monkey, Lawrence rhesus monkey, Tarsius. See, the name spider monkey is because of that. They can put their tail around something and catch hold of it. Camouflage in chameleons is associated with chromoplast, chromosome, chromatophore, chromomere. What is it? Chromoplast is what? Plastid. Chromoplast is plastid, found in plants. Chromosome we know. Chromatophore. Chromatophore. Those are the pigment containing cells in the chameleon. These chromatophores are the ones which will be affected by melanocyte stimulating hormone, MSH, produced by the middle pituitary. Which of the following glands controls egg production and molting in insects? Corpus callosum is a part of the brain. It is the bridge in the brain, mammalian brain. Corpora allata. Corpora allata is what secretes a hormone called juvenile hormone, helps in egg production and molting. Molting is metamorphosis. Molting is metamorphosis, where it changes the stages. Corpora cardiaca also produces a hormone which will regulate molting, but it is not the one which will directly cause molting. Tadpoles can be made to grow in size by inhibiting thyroxine hormone, feeding the eggs of other frogs, injecting gonadotropic hormones, providing them with suitable climatic conditions. Tadpoles can be made to grow in size by what? See, tadpoles are supposed to become frogs. What is the process of transformation of tadpoles into frogs called? What do you call it? Metamorphosis. Metamorphosis is prevented by thyroxine. You give thyroxine hormone, metamorphosis will be prevented. Meaning what? Tadpole remains a tadpole for longer duration. If tadpole remains a tadpole for longer duration, it continues to grow and increases in size before it becomes a frog. Neuroplastic cleavage refers to which type of division of eggs? In birds and reptiles, a portion of the egg divides. A portion of the egg divides with the yolk. That is called meroblastic cleavage. Holoblastic cleavage is the complete egg divides with the yolk. So it is in meroblastic cleavage because only a portion of the egg divides. It is incomplete cleavage. Placenta of human beings belongs to the category of what type of placenta is human placenta? It is hemochorial and deciduate. Hemochorial and deciduate. What does it mean? Hemochorial means made up of chorion filled with blood. Deciduate means it will be shed after childbirth. It will be shed after childbirth. Immediately after ovulation, the mammalian egg is covered by a membrane known as, what is the membrane that covers the mammalian egg? Chorion, zona pellucida, corona radiata, vitelline membrane. And if I change this to amphibian egg, what will be the answer with the same four options? One of the four is the protective acellular covering around the mammalian egg. Similarly, one of the four is a protective acellular covering around the amphibian egg. Acellular covering is living or non-living? Acellular covering is non-living. So which one is for mammalian egg? Which one is for amphibian egg? Corona radiata is present in both mammalian egg as well as in amphibian egg. Corona radiata is a cellular layer. Chorion is an extraembryonic membrane. So it is produced after fertilization and development of the embryo. Between zona pellucida and vitelline membrane, which covers the mammalian egg and which covers the amphibian egg. Zona pellucida is mammalian egg and vitelline membrane is amphibian egg. Vitelline membrane and zona pellucida are similar structures. They are similar structures, alright? Why is it significant? Vitelline membrane. Vitelline membrane is the one which has to be perforated by the sperm. Zona lysine, zona pellucida. Zona lysine is a hormone, which is an enzyme present in the head of the sperm. When the enzyme is released by the rupture of the head, zona lysine will dissolve the zona pellucida and push the nucleus into the cell. Before that, one more enzyme would have acted on the corona radiata. What is the enzyme? Corona lysine. Corona lysine and zona lysine are the enzymes present in the head of the sperm. When it goes, strikes the ovum, ruptures, releases the enzymes, corona radiata will break open all this corona radiata, making way for zona lysine to go and act on the zona pellucida, punctures the zona pellucida, and when there is a passage, nucleus will be pushed into the ovum. Once one nucleus enters, to prevent polyspermy, what happens? Along the margin of the zona pellucida, cortical granules will go and align. So one more sperm comes, it will try to puncture zona pellucida, but it cannot push the nucleus into the ovum because the cortical granules are forming a boundary there. It is forming a boundary on the outside of the cell, inner to the cell membrane. Alright? It's like a human chain. Let's say somebody is being, somebody has to enter this campus, let us assume. They have to enter this campus. Anywhere around the compound wall they can enter. Anywhere around the compound wall they can enter. Only one person is allowed. Only one person is allowed to come inside. Whoever comes inside first, they will be gifted this entire campus. SIRT has given a offer. So people are trying to come from all around. They might be trying to break the compound wall on all the sides. One person breaks it and enters. Now what happens? Inside the campus, closer to the compound wall, there are so many people holding a shield. You see that the war shield they will hold to form a barrier. All of them will go towards the entire boundary of the wall on the inside and stand with the shield. Will it stop the people outside of the compound wall from trying to break the compound wall? It will not stop. There will be another person who will break the compound wall. He will try to come inside. But what happens? Shielded. They cannot enter. The shield, these are all, each cortical granule, consider it as one person holding the shield. They will all stand next to each other, protecting the boundary. Understand? This is how polyspermy is prevented. Polyspermy is prevented by this mechanism. Hominids originated during, hominids are the great apes. Hominids are the great apes, our great ancestors. Hominids originated during the Miocene period. Miocene epoch, not period. Pliocene, human evolution happened there. Oligocene, primates evolved. Paleocene, early mammals. Wings of pigeon, mosquito and bat show what? Pigeon, mosquito and bat. What do they show? Convergent evolution. All of them have adapted to flying, but the structure of them is not the same. Convergent evolution, giving rise to the formation of what type of organs? Analogous organs. They have raised cheekbones, oblique eyes, and yellowish skin colors. Who are they? Who are they? Adrenoids, Negroids, Mongoloids, Africans. Raised cheekbones, oblique eyes and yellowish skin color. Mongoloids, Mongoloid race. Mongoloids are East Asians, Native Americans, Central Asians. The cranial capacity was largest among whom of the following? Peking man, Java ape man, African man, Neanderthal man. Neanderthal man. Peking man and Java ape man were Homo erectus. African man is the early Homo sapiens. So smaller brain size than the Neanderthal man. July 11 is observed as somebody's birthday. What is it observed as? World Population Day. So it is everybody's birthday. World Population Day. Okay. Everybody who is a part of the world's population can celebrate it as their contribution to the commemoration of that day. Okay. All jokers. For a long time, population, see, everything is handled in a short-term approach. Short-term approach. For a long period of time, one-child norm was incentivized. China. Now China has reached its replacement level. Over the next 40-50 years, China will have a lot of aging population, old people. India has the demographic advantage for the next 30 years because 65% of the population of India is less than 15 years or less than 35 years of age. 65% of our population is less than 35 years of age. I am in the 35% of the population. So we have a demographic dividend, demographic advantage, meaning the next 40-50 years can be tremendous growth for India because of the young population. But at the same time, next 40-50 years, there will be decline in the population of China. Japan is already going into decline. So Japan is encouraging people, skilled people, to go and settle in Japan. They want more and more skilled people, knowledgeable people to go and settle in Japan. They are incentivizing more children. Have more children, we will give you rewards. Okay. So, very complex. Resources are limited, human beings are greedy, evolution acts, and along with that there is intelligence also. Evolution drives you to accumulate. Intelligence makes you to plan, strategize, attack, cheat, kill. All of this is evolution. Gone out of hand. No Tobacco Day is May 31st. World Environment Day, I know that, June 5th. World Health Day, April 7th. Cocaine is a stimulant which may cause addiction. It is an alkaloid obtained from erythroxylon coca. Erythroxylon coca. Rauwolfia produces reserpine, which is a very potent hypotensive used for treatment of hypertension. Reserpine. Papaver is our favorite plant, no? Rich plant. Papaver somniferum. What is it? Poppy plant. Produces, what can you get from that? Morphine and opium. Morphine, opium, heroin. Heroin is also processed morphine. If you have some 1,000 square feet of poppy plantation, what will happen? 1,000 square feet of poppy plantation. How you know what is the cost of heroin? Maybe some 2 crores per kg. 1 or 2 crores per kg. So if you just have 1,000 square feet of poppy plantation and produce 1 kg per annum, what will happen? If you have 1,000 square feet of poppy plantation, what will happen? You will go to jail. That's all. Nothing will happen. You will go to jail. Cultivation of poppy is illegal. It can be done for morphine. Morphine is an important painkiller. Poppy plantation is done by proper licensing, which is regulated. Regulated cultivation is allowed. If you consider that as a horticultural activity, then you have problem. Don't do it. A baby has been born with a small tail. It is a case exhibiting miracle. Is it? It is a case exhibiting atavism. Atavism is what? Persistence of an evolutionarily lost character. What has been lost in evolution, if an organism replicates it, if an organism develops it, that is atavism. Metamorphosis we know, change, mutation we know, retrograde evolution, where evolution will take it back to the ancestral state. Mutation can lead to retrograde evolution also. Cyclosporin is used as allergic eczema treatment, immunodepressant, prophylactic for viruses, prophylactic for marasmus. What do you use cyclosporins for? Cyclosporin is a immunosuppressant. In genetic fingerprinting, all of this. All the three are different names for DNA fingerprinting. What is the technique used in DNA fingerprinting? RFLP, STR. What is RFLP? Restriction, fragment, length, polymorphism. And STR is short, tandem, repeats. Short, tandem, repeats. So what do you do? In DNA fingerprinting, you first take a DNA strand, amplify it, create many copies of it by using PCR. then use a particular restriction enzyme to fragment the DNA into multiple pieces. Then what do? You make it single-stranded, do blotting, and then hybridize it with pre-prepared probes, radioactive probes. So wherever the radioactive probes go and bind, that areas will show radioactivity, and radioactivity will be in a particular pattern. So what do? PCR, amplify the DNA, cut the DNA into pieces. Just a moment. Okay, cut the DNA into pieces. Now you have fragments. Make it into, you separate them by chromatography, electrophoresis, gel electrophoresis. So the DNA fragments have moved to different locations on the gel based on their size. Then you make them single-stranded, break them, make them single-stranded, exposing the bases. Then you add, then you blot them onto a nitrocellulose paper. Blotting, what blotting is this? Subtle blotting. You blot it. Now on this paper you have DNA fragments separated based on their size. So whatever, let's say 100 base pair fragment, all the 100 base pair fragments are all in one place. Similarly, different fragments are in different places on the paper. Then you treat the paper with predetermined radionucleotide probes. Let's say 20 base pairs, 30 base pairs, long bases you have prepared. Then when you treat it, these probes are also single-stranded. The DNA on the paper is also single-stranded. If there is a complementary base pair matching for the probe and the DNA fragments, it will go and bind. So wherever these probes have attached complementarily to the DNA, that particular region will start showing radioactivity. Wash off all the excess probes, then you dry the paper, you expose the paper to a X-ray film. You study the paper for radioactivity. X-ray film exposure will give you, film exposure to the X-rays will give you certain pattern, band. Wherever the paper X-ray film is exposed to the X-rays or the rays radioactivity, there will be discoloration. So you get a particular pattern. This has been tested. DNA fingerprinting of a person has been done. This is the technique. Where do we use it? Crime scene investigation. You go to a scene of murder. You find skin scrapings under the nail bed of the victim. Let us say the victim they will fight. No, somebody will not. If somebody comes to kill, and the person says, "Oh, you have come to kill. I don't see any possibility of escape. Kill me." That doesn't happen. They will fight. They will fight and they will try to escape. The most common is nail. When they are fighting, they might scrape off the skin. So under the skin, samples are taken. The DNA is extracted from the sample. DNA is amplified. DNA fingerprinting is made. DNA profiling is done of the sample that was found from the nail bed of the victim. Next, investigation points to 4 or 5 people who can be possible suspects, who could have done the crime. So 4 or 5 people match the bill for the murder. Investigation points to them. Now what do? You take samples from each of them. Do the same process. The key is you should use all the chemicals on these people's samples the same as what was used for the sampling of the victim's nail bed. So the same restriction endonuclease, same medium, same flows. Everything will be same for all these 5 people. Now what will happen? Each DNA profile will give a particular pattern. So you have a pattern that is of the victim's nail bed sample, probably of the killer. You have done the profiling of 5 people who are the suspects. Now if one of these 5 is the killer, or it is the skin scraping of one of these people that was found under the nail bed of the victim, then that person's profile, DNA profile, will match with the test profile that you have created by the sample taken from the nail bed of the victim. DNA fingerprinting helps in this. Paternity disputes. But let's say my DNA profile will be a combination of the DNA profile of my father and my mother. So if my profile is kept in the center and my parents' profiles are kept on either side, my DNA profile will match their DNA profile partially, and collectively this plus this can be overlapped on my profile and there will be similarity. That is the combination that you will find. Now there is a dispute. There is somebody disputing the paternity of a person. A person claims XYZ to be his father. DNA profiling is done. DNA profiling is done of the person who has filed a case, that is the alleged son, mother, and the person whom he is accusing to be the father. Match it. If there is evidence that there is matching of the persons and the alleged father's profile, then he is the father. So for crime scene detection, crime investigation, this thing also. Paternity disputes, identifying unknown bodies, identification of unknown dead bodies. You find some dead body in some place and you don't know who it is. The face is completely smashed. You cannot identify the face. Or a person comes in front of a train completely smashed, no identifiable part left, tissues are left. How do you know who the person is? DNA profiling will help. So DNA profiling is a very important technique. RFLP and STR you should remember: restriction, fragment, length, polymorphism, and short tandem repeats. Short tandem repeats are a particular sequence of DNA which you will find being repeated several times in the genome of a person. STR, short tandem repeats are considered to be unique to a particular individual. Your STRs are unique to you. What is its significance and all that is not known, but STRs are unique to a person. They are like fingerprints. Christian Hansen purified rennet enzyme from calf stomach for large-scale industrial use. Rennet is no longer useful for us. We'll skip it. Lenticels help in gaseous exchange and lenticular transpiration. Lenticels are characteristic of herb stem, shrub stem, woody stem, none of the above. Woody stem. Lenticels are bulgings from the xylem parenchyma, helping in transpiration also. Transpiration is a consequence, but helps in gas exchange for the cells. Anomalous secondary growth is found in out of syllabus chapter. Maximum absorption of water takes place in zone of root cap, zone of elongation, zone of root hair, zone of cell division. Water absorption takes place in the zone of root hair. Tell me the zones from the end towards the plant body. What is at the end? Tip, root cap, then zone of root hair, then meristematic zone, then you have elongation zone, then you have maturation zone. The first part of the maturation zone is your root hair. Next. The most accepted theory of ascent of sap is transpiration pull theory, Dixon and Jolly. Jolly LLB. Have you watched it? Okay. Did you watch it here? Did you watch that Lucky yesterday? Which movie did you watch? Did watch the movie. Right? So studying, relaxing in the room. What were you all doing? Huh? Wi-Fi was not working. Why Wi-Fi? You can download it from mobile, transfer it to pen drive, play using pen drive. Movie time should not be missed. Important. Okay. In 1971, Theo Diener discovered a new infectious agent that was smaller than viruses and known as viroid. Which of the following diseases is caused by viroids? Which of the following is a viroid disease? PSD. Potato spindle tuber disease is a viroid disease. Bunchy top of banana apparently is a viral disease. Witch's broom is also not a viroid disease, it's a bacterial disease. Viroids are the smallest known pathogenic or infectious agents. What is the smallest known pathogenic living organism? Mycoplasma. The bacteria without cell wall. What is common among diatoms, desmids, dinoflagellates, euglena, trypanosoma, plasmodium and slime molds? All are prokaryotic in nature. Are they prokaryotic? All of them are eukaryotic. All have non-histonic proteins and lack histone proteins. No, histone protein is common to all DNA. All are single-celled eukaryotes which come under kingdom Protista. That is true. All of them are protistans, single-celled protistans. All are multicellular eukaryotes which come under kingdom Fungi. Diatoms and dinoflagellates are photosynthetic algae. Euglena is mixotrophic, both autotrophic and heterotrophic. Trypanosoma and plasmodium are parasitic. Slime molds are fungus-like. Trypanosoma and plasmodium are parasitic and also pathogenic for humans. What is the disease they cause? Trypanosoma causes sleeping sickness. Plasmodium causes malaria. So kingdom Protista is an artificial group of organisms which are unrelated to each other. Slime molds are parasitic protists which form plasmodium. Is it correct? Is the statement right? Is anything right in the statement about slime molds? They do form plasmodium, which is a protective mechanism for survival, but they are not parasitic. Slime molds are saprotrophic. They are saprotrophic. It is slime molds which will eat away your old photographs, paper. Okay. Have you seen old photographs of your great grandparents or some old photographs of 50-year-old? With the frame only, there's boundary all eaten up. Slime molds. Walls. Not slime mold, that is moisture removing the paint. Saprophytic protists under suitable conditions form plasmodium. That is correct. They are not parasitic at all and they are not monerals. Which of the following is not correct about slime molds? No, we did that. Same options, no? Now there are different questions, different options. Which of the following is not correct about slime molds? Under suitable conditions, they form plasmodium. Is it right or wrong? It is right. Under suitable conditions, plasmodium is formed. Under unfavorable conditions, plasmodium differentiates and forms fruiting bodies bearing spores at their tips. That is also correct. When the conditions become unfavorable, plasmodium forms the fruiting bodies, spores are produced, dispersed, trying to increase population and survivability. The spores of slime molds lack a true cell wall. Is it right? The spores of cell wall do have a resistant cell wall. They don't lack a cell wall. They have very resistant, tougher cell wall. So that will help them survive harsh conditions. Why? Spores are produced under unfavorable conditions. When the conditions are unfavorable, plasmodium starts developing the spores and dispersing it. So when the conditions become favorable, the spores have to germinate and grow. Till then they have to survive, and it is the tough cell wall which will help them, resistant cell wall. Which of the following steps is not involved in the sexual life cycle of fungi? Fusion of male and female gametes results in prothallus. Prothallus formation does not happen in fungus. Prothallus formation occurs in pteridophytes. Okay. Fusion of male and female gametes results in prothallus is wrong. Fusion of protoplasms between two motile or non-motile gametes is called plasmogamy. Is that correct? That is correct. Fusion of two nuclei is called karyogamy. Yes. Meiosis in zygote results in haploid spores. That is also correct. Statement 1 is wrong. So there are 3 stages involved in the sexual reproduction of fungi: plasmogamy, karyogamy, and meiosis. Plasmogamy, karyogamy, and meiosis. Which of the following groups represents the membrane of the same members of the same class? Mucor, Rhizopus, Penicillium. Mucor. Oh, this is going deeper into fungal classes. Mucor, Rhizopus, Penicillium. Mucor and Rhizopus are ascomycetes, zygomycetes. Penicillium is ascomycetes. Yeast and Penicillium are ascomycetes. Albugo is oomycetes. Aspergillus, claviceps, and neurospora. All of them are ascomycetes or sac fungi. Different types of fungi: sac fungi, club fungi. Right, club fungi is basidiomycetes. Okay, ascomycetes is sac fungi. So C is right. Aspergillus, claviceps, and neurospora are all ascomycetes. Agaricus is basidiomycetes. Coletotrichum is ascomycetes. Agaricus and Ustulago are basidiomycetes. What is the basis for classification of fungi? Structure of the thallus or structure of the mycelium. Mycelium structure, then mode of reproduction, then spore production. Which is the class of fungus that does not show sexual reproduction? Deuteromycetes. Oomycetes, zygomycetes, ascomycetes, basidiomycetes. All 4 of them show sexual reproduction. Deuteromycetes do not show sexual reproduction. In fungus, Deuteromycetes is considered to be like a temporary place until you find where they belong to within fungus. So Deuteromycetes is like protista within fungus. Which of the following is not correct about Deuteromycetes? Okay. Alternaria, Piccinia, Coletotrichum, and Trichoderma are members of Deuteromycetes. Oh, these are difficult questions. You have multiple examples. Let's see if there are any sensible options for us to arrive at conclusion. We need to find out one wrong statement about Deuteromycetes. First one, examples are there. Coletotrichum, Alternaria are all Deuteromycetes. Alternaria, Coletotrichum is ascomycetes, as we saw in the previous question only. Okay, let's see option B. Deuteromycetes are known as imperfect fungi because only the asexual or vegetative phases of these fungi are known. That is correct statement. They are imperfect fungi. Deuteromycetes reproduce only by asexual spores known as conidia. That is also true. Many members of Deuteromycetes are transferred to ascomycetes or basidiomycetes. Right now we discussed. They are like temporary house for them. Once they are identified, once their life cycle is properly identified, the fungi in Deuteromycetes can be pushed into respective classes. So statement 1 is wrong. So Piccinia is basidiomycetes, not a Deuteromycetes. Others are Deuteromycetes. Colletotrichum is ascomycetes. Yes. Next. Which of the following is not correct about algae? Lot of questions related to properties and examples in this paper. That is why I think many of you have got response in this paper. Okay. Not correct about algae. Porphyra, laminaria and sargassum are edible algae. Is it right? It is correct. Agar-agar, which is used to grow microbes in culture media, is obtained from Gelidium and Gracilaria. That is also correct. Algin is obtained from red algae, while carrageenan is from brown algae. What is right about it? Reverse. Algin is obtained from brown algae, carrageenan is obtained from red algae. That is wrong statement. Chlorella and spirulina are unicellular algae rich in protein. Correct. What is the kind of protein that you extract from chlorella and spirulina? Single cell proteins. Single cell proteins. Advantage of single cell proteins? Very rich in essential amino acids, easy to digest, very good bioavailability. Which of the following groups represents all members of Phaeophaceae or brown algae? So we have to find out one of red or green algae in the wrong options. So try to figure out one red algae or green algae in the options. Spirogyra is green algae. Spirogyra is green algae. Gracilaria and Gelidium, which gives us agar, they are red algae. So in the Gracilaria, Gelidium, remove it, D removed. Laminaria. Laminaria is brown algae. Spirogyra is green algae. So A and D ruled out. A and D ruled out. In between B and C, Dictyota, Polycyphonia, Porphyra. Polycyphonia and Porphyra red algae. So C is also ruled out. All members of Phaeophaceae are B. Ectocarpus, Laminaria and Sargassum. What is the pigment in brown algae? Pigment in brown algae. Huh? What gives it brown color? Chlorophyll A is common to all three: green, brown, red. B, C, D is what? Green, brown, red. So, other than that, what gives brown color to brown algae? Fucoxanthin. What gives red color to red algae? Phycoerythrin. The predominant stage of the life cycle of a moss is sporophyte, gametophyte, triploid, diploid. Moss is what? Bryophyte. Dominant is the gametophyte and dependent is the sporophyte. So when gametophyte is the dominant form, what is the ploidy? Ploidy of the gametophyte is haploid. A moss which provides peat moss that is used as fuel and for packaging material for plant stays. Which one is peat moss? Sphagnum. Sphagnum is peat moss. What is the advantage? It is used as a soil conditioner. It is used as a soil conditioner. It can absorb several times its own weight as water, several times its weight of water. So water retaining properties, peat moss. Okay, A and B diagrams are given. Steel region of dicot and monocot roots are given. Identify the correct difference between the two, A and B, in the choices given. Pith is absent in which stem? Which root? Pith is absent in monocot root. Pith is present in dicot root. So, correct answer Absence of pith in A and presence of pith in B. That is correct. Xylem, you look at it. Tetrarch xylem and polyarch xylem. That is the combination that we have to look at. A is tetrarch xylem. Tetrarch xylem is four xylem bundles. B is polyarch xylem, many xylem bundles. So dicot roots do not have dicot roots. Pith is absent in the dicot root. Question number. See that large pith in monocot and obliterated pith in dicot. Okay. Monocot is polyarch, dicot is tetrarch. In pteridophytes, spores germinate to give rise to an inconspicuous, small, multicellular, free-living, photosynthetic, thalloid gametophyte called, what do you call this gametophyte in pteridophyte? Prothallus. Prothallus. What is protonema? Protonema is the gametophyte stage in bryophytes, not pteridophytes. Archegonia is the female reproductive organ. It is not the entire gametophyte as such. Ovule is of the seed plant. Pteridophytes do not have ovule. The female gametophyte of angiospermic plants is called. The female gametophyte of angiospermic plants is called what? Embryo, embryo sac, agapparatus, synergics. Which one is the gametophyte as such, if you want to call it? Embryo sac. Embryo sac. In angiosperms, each embryo sac has a 3-celled agapparatus, which contains 1 egg cell, 2 synergids, 3 antipodals and 2 polar bodies, all of this. Option B, all of this. All of these are present in embryo sac. Quickly tell me who will fertilize the egg? Who will fertilize the egg? Male gamete. Who will fertilize the antipodals? Nobody. Who will fertilize the synergids? Nobody. But male gamete will break it over. Who will fertilize the central cell or the polar nuclei? Male gamete. There are 2 male gametes. Largest percentage of molecules that make up the cell membrane are. Largest percentage of molecules that make up the cell membrane are. What is the cell membrane made up of? Predominantly phospholipid. Phospholipid. Anything without phospholipid has to be ruled out. There is only one of 2 other organisms. Keynote species, keystone species. Keystone species, we already discussed. Species which are crucial for the survival of the ecosystem. They play a supportive role for the other species. So keystone species are essential for ecosystem stability. The complexity of what determines the stability of an ecosystem. What what are the factors that will determine the stability of an ecosystem? Stability of an ecosystem means that it can survive by itself. You don't need to provide external support for it to survive. What is it that will provide stability? Complexity of the food chain. Okay, partially correct. Every species should be able to survive and thrive. That is the necessity for a stable ecosystem. Correct? Not the food chain. There should be more food chains with intricate connections between them. What do you call that when multiple food chains are connected to each other? Food web. A complex food web makes the ecosystem stable. So nothing is lost. Predators will find something very easily available, they will start hunting that, eating that. When the number of that particular thing starts reducing, it becomes more work for the predator to catch hold of that. So they leave it and catch hold of something else which is easily available. So the one which was reduced in numbers get time from the predator's attack to multiply further. So it's a cyclical process. What are indicator species? What are indicator species? There is one very important indicator species which will tell you about environmental pollution, air pollution. Lichens. Indicator species are those species which will give you signals about something being wrong. Lichens will not grow in polluted environment. Lichens grow in non-polluted environment. So presence of lichens in a particular area is a proof that the place is not very polluted. Refer to the given figure on photoperiodism and select the correct option. 121. ABC. A. No correlation between light period and flowering. B. Long light exposure period. C. Short light exposure period. See, what does this indicate? A, B, C, all three show you one phase where there is flowering and there is flowering. So, A has, when you see the flowering, it means that there is a period of no flowering also. So when the flowering is not found, that is the time of exposure which will lead to the flowering in the next phase. So what is A? Light exposure. What does A talk about light exposure? Short light exposure. B is long light exposure. C is no correlation between light period and flowering. So A is a long night plant, B is a long day plant, C is a day neutral plant. If the DNA codons are CAT, 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 if the DNA codons are cat, and the guanine base is added at the beginning, Which of the following would result? C80, guanine. A frame shift mutation. GCA, TCA, T. What will happen? If G is added, if a guanine base is added, GCA, TCA, T is correct, no? C. One base is added. So you are shifting the frame. Addition or deletion of bases will lead to frame shift mutation. Substitution will lead to point mutation. Mutation in two types: point mutation and substitute and frame shift mutation. Here addition. Where is the addition happening? At the beginning. So what is the right choice here? Both B and C. Frame shift is also happening and the codons are also changing. Following DNA sequence occurs at the start of a DNA strand. Which of the sequences below would most likely bind to this sequence to initiate DNA replication? See, look at the polarity. Complementary sequence should be 5 to 3. The given strand is 3 to 5. Complementary sequence should be 5 to 3. So you should look at the sequence complementary from 5 to 3. Check for it. Look each option at the 5' end. Is it complementary to the first 2 or 3 bases? First 3 bases in the 3 to 5 is given sequence is AAT. Complementary to that should be TTA at the 5' end. Complementary to that should be TTA at the 5' end. Look at option A. At the 5' end you have UGA. Rule out. Option B, at the 5' end you have UUA. Rule out. Remove. Don't look at more bases. Just check for 2 or 3 bases. TTA at 5' end is option D. Option C at the 5' end is TGA. That is also ruled out. So without looking at the entire sequence, you are looking at 3 bases at the right polarity and cross-checking with it. That's all. These are time waster questions, but if you approach it like this, that we did now, you will save time. Otherwise, if you have to match every base and go on, it will not work out. You can do that with the other end also. You can do it with 5' end. Look at the 3 bases. What should be the complementary sequence? Check for the complementary sequence on the 3' end. Are you understanding? Did you understand the logic there? No. Which of the following enzymes is correctly matched to its function? Helicase stabilizes single strand DNA. Is it correct? Wrong. Helicase role is to break the double strand. Stabilization is done by a protein. What protein? SSB protein. Single strand binding protein. Topoisomerase breaks bonds between nucleotides. Is it correct? Topoisomerase. Topoisomerase is unwindase. So it doesn't break bonds as such. DNA polymerase 3 adds nucleotides to the lagging strand. Is it correct? That is correct. DNA polymerase 1 closes gaps between Okazaki fragments. DNA polymerase 1 closes gaps between Okazaki fragments. DNA polymerase 1 removes the RNA primer, but its job is not to seal Okazaki fragments. That Okazaki fragment sealing is done by what? Who seals it? Ligase. DNA ligase, not polymerase. DNA polymerase 2 does what? What is the role of DNA polymerase 2? Proofreading. So DNA polymerase 3 will build the nucleotides. At multiple places you have primers. Polymerase one will come, remove the primer and add DNA bases there. Polymerase 2 will come, cross check for correctness. If it is all correct, it will pass. Ligase will come, do the soldering. Why is ATP such a good energy currency molecule? It is very stable. Its negatively charged phosphate groups repel one another. It has 3 phosphates that repel one another, forming a stable molecule, forming an unstable molecule. It has double covalent bonds which store twice as much energy. ATP is a good currency molecule because the bonds between phosphates is high energy bond, but also because of the repulsion from one another, it is easy to break. You want the bond to be a high energy bond, but the bond should be easily breakable so that you can break it and use the energy when you require it. Both characters are fulfilled by ATP. Which of the following factors is not involved in determining whether light is absorbed by a molecule or not? What does not influence light absorption is the question. Chemical structure of the molecule, does that influence whether light is absorbed by a molecule or not? It does. Wavelength of the light, is it important? Wavelength of the light is important for absorption. Intensity of light. Intensity of light is a function of number of protons per unit area that the light is passing through. Wavelength and frequency need not be different. If more photons are passing through a particular cross section in a given unit time, then it is of more intensity. Intensity of light does not determine whether absorption happens or not. Absorption happens based on the wavelength. Potential energy levels of electron shells in the molecule. What is the evidence for the photosystem? Chlorophyll absorbs light of 2 different wavelengths, blue and red. Chlorophyll absorbing 2 different wavelengths, blue and red. Is it an evidence for 2 photosystems being present? Can we prove it? No. The combination of light at 680 and 700 nanometer is much more effective at stimulating photosynthesis than either wavelength alone. Now see, blue spectrum and red spectrum both are being observed. How do we know whether it is in 2 different photosystems or it is in a single photosystem? Then when you use a 680 nanometer wavelength light, photosynthesis happens to a certain extent. When you use 700 nanometer wavelength light, photosystem happens to a certain extent. When you use a combination of both 680 and 700, then the photosynthetic output is much more than using them alone. This is an evidence of 2 systems operating in this process. Microscopy reveals 2 colors of chloroplast within the same leaf cell. Now, 2 different high energy molecules are produced during the light dependent reactions of photosynthesis, ATP and NADPH. That also cannot justify that there are 2 photosystems. Binomial system of nomenclature uses 2 words for a name denoting. What is binomial nomenclature? What does it denote? Species and genus. Who devised it? Carolus Linnaeus. So which one do you write in capital letters? Genus name. Italics. Both are to be written in italics. Species name is written in lowercase. Which of the following are the two growth forms that fungi have? Mycelia and chytrids, chytrids and zygomycota, yeasts and mycelia, hyphae and mycelia. Yeast is unicellular form. Mycelia is mycelia are multicellular filamentous network of hyphae. Those are the two growth forms that fungi have. Which of the following is a phyllodes? Eichhornia, Parkinsonia, Passiflora, Opuntia. Opuntia is spine, modified leaves modified as spine. So Opuntia is not a phyllodes. Eichhornia is a water plant, no? Eichhornia is a water plant. So it has air-filled petioles. Yeah, leaves have air pockets, making it floating. Petioles are air-filled. So Opuntia and Eichhornia ruled out. Between Parkinsonia and Passiflora, it is Parkinsonia which has a phyllodes. Phyllodes is also a modified petiole. Phyllodes is a modified petiole. Functions like leaf. It is it has chloroplast. G1 phase of the G2 phase of the cell cycle is characterized by the presence of. Do we have normal diploid chromosome during G2 phase? See, this has some options which G2 phase of the cell cycle is characterized by the presence of normal diploid chromosome. Is it right or wrong? Let's look at the other options. Single chromatid. Is it right or wrong? Wrong, because you will have two chromatids for every chromosome. Abundant RNA without any difference in the amount of DNA from the S phase. Right or wrong? Abundant RNA is present. In the G2 phase, there is a lot of protein synthesis happening. But is the amount of DNA no different from that of the S phase? You are not comparing here with the parental cell. You are comparing here in terms of the amount of DNA with the cell at S phase, after S phase, which is same, no? DNA doesn't change. The amount of DNA in G2 phase will be the same as that was at the end of S phase. But here, both the cases, the DNA content has become double. DNA content has become double. Chromosomes, normal diploid chromosome. Why is it wrong? Option D is also wrong. Double the amount of DNA than present in the S phase. That is wrong. It has the same amount of DNA as present in the S phase. So option C is correct. Presence of normal diploid chromosome. Is it right? It is not characteristic, but amount of DNA present in the space, double amount of DNA is also not nothing to do with G2 phase. G2 phase is preparation again. See, now look at it. In G2 phase, the DNA replication has happened. DNA replication has happened. Every chromosome has a duplicate copy, so they are in the form of a chromosome with two chromatids. But the number of chromosomes will be same, no? The number of chromosomes will still be same. So why should not we consider that G2 phase cell has normal diploid chromosome? Why not? It still has normal diploid chromosome only. Though the DNA content has increased, chromosome number has not increased, no? Neither increased nor decreased. So option A is also not wrong. Between A and C, C is a better option to choose. It goes more detail. It goes into more detail. Identify what is true about anaphase 2. Don't look at the options. Tell me what happens in anaphase 2. Separation of sister chromatids. Separation of sister chromatids. Now let us look at it. Okay. It is the second meiotic division. Sister chromatids separate. Homologous chromosomes separate. Division of centromere occurs. Sister chromatids separate. Doesn't the centromere also split, divide? Isn't there splitting of centromere as well in anaphase? So why is D wrong? D is talking about a process which will lead to the separation of sister chromatids. So separation of sister chromatids is a better choice there. Homologous chromosomes separate when? When do homologous chromosomes separate? Anaphase 1. We'll do till 1:35. During meiosis, crossing over occurs between non-homologous chromosomes, between sister chromatids, between non-sister chromatids, none of this. Between sister chromatids, non-sister chromatids. The statement is incomplete. Complete it. Between non-sister chromatids of homologous chromosomes. Between non-sister chromatids of homologous chromosomes. That is the right choice. Colchicine interferes with cell division by, what does it interfere? Colchicine blocks the spindle fiber formation. Colchicine blocks the spindle fiber formation, microtubule formation, preventing spindle fiber assembly. Colchicine can be used for inducing what? For inducing aneuploidy. The wheat that we use is hexaploid wheat. The wild wheat that is found, that is the original wheat, is a diploid wheat. How did we get hexaploid wheat? Polyploidy can be induced by treatment of the gametes. When the division is happening, you treat the reproductive cells with colchicine. Disjunction of gametes will not, disjunction of chromosomes will not happen. So the entire set will go into one chromosome. You have a diploid male gamete and a diploid female gamete fused to form a tetraploid progeny. Now tetraploid progeny can be made to combine with a diploid mate to get hexaploid. That is how the wheat that we eat now has been produced. Hexaploid wheat. Colchicine. DNA that is transcribed to mRNA is antisense strand, sense strand, both sense and antisense strand, parts of antisense and sense strand. Now, antisense strand, sense strand. There are two strands given. Which of these is the coding strand? Sense strand is the coding strand. Which is the template? Antisense is the template. Now look at the question. DNA that is transcribed to mRNA is, should we consider what the mRNA is a copy of, or should we consider it as what is the basis on which the mRNA is getting transcribed? So transcription is creating a copy. Transcription is creating a copy against a template. So DNA that is transcribed to mRNA is transcribed by using the template strand as the template. The template strand is antisense strand. Alright? So if you consider transcription as a process, antisense strand is the template. If you have to consider antisense strand as the choice here. Right. Enough for today. We have done about, we have quickly run through about 70 plus 35, 105 questions. We'll break for lunch.